okay, and then where's my continue? Okay, okay. So once more. So welcome to the Advanced Cytoscape workshop. Uh, this is a pretty experimental setup. I'm trying to run it this time in a pretty interactive way. So in order to participate in a reasonable way, you need a few things installed. I did paste a bunch of links into the chat, but I'll go through those things once more here. First, you do need to have a cytoscape installed. Here's the link. I'm gonna paste it into chat once more. Okay, and you've got to go to download if you didn't do it before, and then depending on what operating system you're gonna run, you'll have to click, and this thing should in a pretty transparent way get itself installed. So this is one part that you need for today. You will also need a few files with the data needed to create to create a network we're gonna play with today. So the link is, the files are here. This is under day one, and the link is just going into chat. Okay, here's the link to the directory with the files. My suggestion is you take all those files you see in a directory and you place them in a directory in your desktop, so that you can keep track of those. So those are two things that we need for today. For tomorrow and Thursday, we will write a little bit of Python scripts. To do this thing, my suggestion is you install Anaconda. You can do it later today. This might take a while to get in. I think the files relatively large. You go to Anaconda site. Here's the link in the chat, and then you go under products, individual edition, and then down here there are downloads, and depending on your operating system, you install one of those three things at the top. We're gonna use Spider out of the entire Anaconda package. So if you do have Spider installed, then you can skip this step. But if you don't have Python installed, getting Anaconda is pretty convenient one-stop way to get going. As far as Python goes, we're gonna use very basic constructs. So I think prerequirement for this workshop was intro to Python. If you didn't participate, then if you've got a little bit of experience with scripting in Python, you're gonna be fine. For your reference, here's a link to, let's go here. Uh, Here's a link to, where is it? Something's messed up. Let's see. Here and here. Here's a link to the directory with whatever was covered in the intro to Python. And if you click through those day one, day two, and day three files, you're gonna essentially see what was covered in a Python workshop. The cheat sheet here essentially lists all the constructs that all the all the Python bits and pieces that we went through in that workshop. So this is maybe 3-4 pages of basic Python. So this is for your reference. If you think you're comfortable with what you see in those files here, you should be fine. So there's nothing to be scared about. If you never scripted, you might get stuck, but I'll try to go slowly and you might survive. So we'll see how it goes. So those are essentially prerequisites. And then one more thing, if we go back to advanced cytoscape and winter, there's also this set of slides in PowerPoint. Those are covering more or less the experimental, the biology or molecular biology or biochemistry background for protein interactions. This will pop up tomorrow when we'll go over the interaction records and what's in there. The PowerPoint slides do show up a little bit of information on where the molecular interactions, where the interaction records are coming from, how interactions are discovered, and this is normally covered in an introduction to Cytoscape workshop that was run earlier this year in a fall. So you might want to just click through those slides. Don't memorize. Just see what's there and use it as a reference. Okay. So those are the. This is the background. Let's go to Cytoscape. Assuming you installed it, you should be able to run it by either finding an icon. You should see an icon showing up somewhere on your desktop, or you might have, in case you're on Windows, you might have to go to Programs, and then Cytoscape should show up here, and then you can run it here. It should start after a while. Okay, here we go. And here's the initial startup screen. It's doing something strange. Okay, it's initializing. The first time you run it is gonna take more time because it's trying to initialize things, get things in from the. There's no. Oh, those two slides. Just a moment. Let me find it. Slides are here. So here, copy, and here's the link to slides. Okay. So in the meantime, Cytoscape started. Okay. And that's how it looks like when you pop it up. Cytoscape is pretty modular. There's a lot of additional modules that can be loaded. They are called apps, and initially you're not gonna see too many things listed down here under apps, but if you go to App Manager, then what we'll need is the app called Bingo, this guy. So you'll have to click on it. Okay, I'm gonna uninstall it. Okay, and then, so when you start it, close. Okay, so initially you're gonna have apps, and you go to App Manager, you're gonna select Bingo, and then you're gonna install. And then once you're done in the apps, you should see Bingo listed as one of those. So that's our initial setup for the Cytoscape. Okay, now Cytoscape is a program that is used for showing networks of various sorts. Uh, in general, it's pretty agnostic of what the networks are, what the graphs are representing. So it can be used in many different contexts. In our case, we're interested in biological, in molecular biology applications. So the networks would be corresponding to, let's say, interactions between proteins, or interactions between proteins and DNA, or things things like that. Where do they come from? Well, in a directory that I've got here, I do have a few files that can be used to create interaction networks. Let me get those 3 files out into a directory, so I can get them from my desktop. So let's try. Can I select everything? And now let's see if I can drag it. Can I drag it up, down? Let's do it this way. Upload. Can I? Oh, this is not the best way. So let's cancel. Okay, I'm gonna make myself a directory. Let's make myself a folder. Let's call it PPI. Okay, here, and then let me drag those files one. Can I drag it? No. Ooh, that's annoying. No, I can't. Okay, so let's download and save. Okay, I'm gonna save. I'm just saving all those files one by one. Okay, and save, and save, and save. Okay, something happened. I don't want this one. Okay, and now they ended up somewhere in my download. So now I'm gonna take all of them and I'm gonna stack them into this directory in a desktop. So now I've got those files here. Well, I'm missing this one, so I'm gonna do the same thing. Download and save, and once more here, and this one is gonna go here. Okay, so now I've got exactly the same file as in Google Drive sitting in my desktop in a PPI directory. Okay, so now let's try to look at those. I'm gonna look at the file called galfilterededge.txt. Okay, and I'm gonna open it. Let's say, let's do it with Notepad here. So here, the background is horrible. Let's see, settings, preferences, general. I'm gonna, okay. So here's our get filtered edge.txt file. It's got 3 columns there. Okay, the first and the last one are listing identifiers of the proteins or genes that we're interested in, and the middle column is providing information. Let me get this thing bigger because the font is horrible. I'm not gonna tools, settings, preferences, editing new, no, tools, settings, style configurator. Let me, where do I set up? Okay, here, let's make it. Why is it? File, settings. Can I document? No. Why can't I make the font bigger? Tool settings, style. Okay. Okay. Default. Yes. And settings style. Okay. Okay. Save here. That's better. Okay. So the file lists identifiers or proteins in the first and third column, and in the middle column there's information about what's the type of interaction between those two proteins we're dealing with. Convention is that if the both molecules on the left and on the right are proteins, then the type is PP. If the molecule on the left is protein and the molecule on the right is DNA, meaning that this is a protein that binds to the gene coding for this protein, then the code is PD. P corresponds to the left molecule, D corresponds to the right molecule. So this is interaction between protein binding to a gene corresponding to YJR, whatever the gene is. Here we've got the protein with this identifier binding to a protein with that identifier. So this is essentially a list of pairs of either two proteins and protein and the gene, and that interact. So that's what in our file is. When we to open it in a Cytoscape, you go just to File, you go to New, you go to Import, you import network from File, and now I'm gonna go to my desktop. I'm gonna go to PPI, and I'm gonna pick up this edge txt file. Okay, so now once I open, I do see initial import screen. At this moment, the file, the columns are not, the file is not split into columns. To make it split into columns, we go to advanced options. Here, we split on a space instead also to have now 3 columns here. and we are not using the first line as names because we didn't have titles for the columns. So we're just gonna leave this thing unchecked. We start importing from first row, and this is okay here. And now we've got to specify that the first column is a source node. This is one end of the interaction. We've got to specify that the third column is the other end, so target node, the other end of the interaction. So this is where one side of interaction is. This is where the other one is, and then the column 2 is gonna be our interaction type. Okay, so now once we've got this thing, we can now just click on OK, and here's the interaction network that's described in this file. As you notice, there's nothing special about the file. The only thing it contains is 3 columns. There's a one column with protein identifiers. There's a column specifying what type of interaction we're dealing, and there's a third column specifying the other side of the interaction. So files like this can be prepared in Excel. File like this can be prepared in a text editor. Files like this are very simple to create with any scripting language. We'll play with this thing over next 2 days with Python. But there's nothing special about preparing those files, provided we know what to put there. So this is the most complicated part, actually, to figure out how to get to information that allows you create files that Cytoscape can deal with. But otherwise relatively easy. And once we've got the file, we can create create interaction network. Basics of navigation, how to look at those things. We can move around by just dragging with the left button pressed. We can move the network around. If we scroll, if we use a scroll wheel on a mouse, we can zoom in and out. We can also zoom in using the icons here. Plus is gonna get us closer to the network. As you see, when we get close enough, we start to see the stickers on individual proteins there. We can zoom out with the minus. We can, if we want to see entire thing, we can click on the icon here. It's gonna get you entire network in the view. We can select nodes by holding Control and dragging a rectangle here. Then we can select the part of the network. If you want to zoom on a selected part, we can click on a fixed selected icon, and then things that were selected will get shown in the window. The rectangle on the right is a view of entire network with the rectangle corresponding what's visible in the screen, so we can just drag things around and move around the network to whatever region we're interested in. So that's the basics of moving around. If this does not modify the network, in a sense that we are not modifying the shape of the the position of the nodes. We don't change the distances between the nodes. All we're doing is we're just getting things. We're getting to the network closer, and we move it around. Okay. To modify the network, we can use the layout tools here, and this allows us, let's go back here, this allows us to rotate the network. There's a slider at the bottom, and we can rotate what shows on the screen. Okay, and we can now zoom in a little bit here. And as you see, when I'm zooming in and out, the relative size of the labels of the nodes and the distances between those don't change. This is just zooming in, getting closer, farther away. To change the distances, to scale the size of the network while keeping the size of the stickers the same, we can go to layout tools and then use the scale slider, and then we can make the network bigger. You see that now the stickers don't overlap anymore. Before we were somewhere here and some of them were overlapping. So this is the way to space things farther away so that they don't obscure the view. So this is different from scaling. With scaling, the relative distances between nodes and the size of the nodes don't change. So those are two differences. Going back to our view, so that's our network. Now, so far there's nothing there except for information who's connected with whom, and we can either and we can look at that by just making observation that the node called YDR395W is connected to YPR102C. That's what's seen in the graph. We can also get to information about those elements in a tabular form. At the very bottom, below the graph panel, there's a panel that shows tables, and there are three tables. There's a node table, there's an edge table, and there's a network table. The last one is, for us, least interesting. At least different tables, different networks that might be present in the Cytoscape at the same time. But if we look at the edge table, we're gonna see information about the network we've got here. It's very similar to whatever we've seen in a file. There's gonna be a column with a shared name, and this is the name of the interaction. There's gonna be interaction column with the interaction type. This is this column here, which lists the middle column, corresponds to the middle column in our file. And then there's information repeated as a name and interaction. The difference is those two columns are used for the names visible in this particular network. In case we've got several networks, each using the same protein, displaying the same interaction, the shared name is referring to the common name between all those networks. So those, for our purposes, are exactly the same. Now, this is the table which shows us everything, all the interactions there. If we want to see something we're interested in, we can always select interaction or a group of those. Once we select it with control and dragging marquee, we now selected several several interactions, and now the table shows only selected lines from the table. So this way we can easily find what's in a table for the interactions, and the same thing holds for a table with the nodes. Now we've got 1, 2, 3, 4, 5 nodes selected, and here are the 5 rows in a table. At this moment, it's very uninformative. It only gives us the identifier of the protein. So there's nothing that interesting here, except that if we don't have anything selected, the table is going to show us everything. If we want to select one particular edge, one particular interaction, we can click on it if we're lucky, and then we select only and see only this particular interaction in a table. Same thing holds for nodes. If we select a node and go to node table, we do see only the row corresponding to the selected node. So this is the way to quickly find information about one particular, or let's say 3 particular proteins or molecules or whatever's at each node of the network. So that's how a network can be read from a file. So far, there's nothing there but description of who's connected with whom. To get more information, we can utilize files that look like, let's see, that look like, where I am, desktop and PPI. Okay, and let's get the file gal filtered node txt here. So let's open it. Now this is a file that's quite typical of many types of experiments that might be performed. In this particular case, it's taken from a on a Mac. Let me check, but I'll have to. Okay, I've got to log in into Mac 2529, unless there's someone who knows. Let's go to Cytoscape. Well, how do I select? That's a good question. Let's see. I can just press on a scratch pad and it selects. This is for one guy to drag. Let's see. Not control command. No, that's a good question. No. Well, if nothing helps, we can always go to help. Let's see. Can go Cytoscape. Mac navigation user on Mac Okay, zoom. I'm not a Mac person, so I do have, okay, zoom in, selected pan. Let's see, zoom, zoom, select, let's see. Oh, someone. Okay, there's a now. Thank you very much. I'm not a Mac person. Okay, so going back, going back to the file we've got here. This is a file that represents typical results that are of experiments that are shown in Cytoscape. In this particular case, it's a table that lists individual genes and then the level of expression under different conditions. Okay, there are 6 columns here. There's one condition and the level of expression. This is actually log ratio of expression in a given conditions relative to some reference state. So that's the number which tells us if expression went up or down relative to control. So positive numbers mean expression went up, negative it means the expression went down. And then the second column gives us estimate of how significant those changes are. You can imagine that if both expression levels in a given conditions and control are high, then the changes are more reliable to report. So the significance is gonna be higher if the levels of expressions are very small, then our the significance of the change might be smaller. So this column here lists how significant the changes are, and there are 3 sets of those for three different conditions. So this is a table that typically is coming out of experiment of some sort, and this is information we want to overlay on an interaction network. So let's try to get this thing in. Okay, to get the information in, we've got to go to file. I'm gonna get this thing back to, and I'm gonna go to file, import, and I'm gonna now import table from file. To get the network, I was getting network from file. Here I'm getting table from file. Okay, I'm gonna pick up get filtered edge txt. That's not ht. I'm gonna get filtered node txt. Okay, and now I've got the import window similar to what we've seen before, except that we've got to specify a few other things. Okay, I want to import to a network collection, so we've got to get it to everything here. This doesn't change. We are importing data as a node table columns. This is because we're getting lines, rows corresponding to different proteins. So those are corresponding to annotation that goes on the protein. So we're selecting node table. Okay, we use case sensitive. If we want to, we can leave it. And now we've got to specify what we're checking. We've got to check what's gonna go in. Okay, one column in a file is gonna be so-called key column. This is the column that contains names identical to whatever is used for naming the protein, the node. So in the network, our names used for naming the nodes look like YMR043W. So those are the standard names of ORFs or of genes in Saccharomyces, and this is our key column. And then apart from that, we do have several column 6, as we've seen in a file, each one corresponding to column of numbers, and we can check that we indeed are getting those as numbers. There are different options. It is possible to force a column to be a string instead of a floating point number, or it can be made into integer or just into yes and no values into just Boolean variables. So we've got a control of how the columns got in. In most of the cases, Cytoscape is gonna figure it out automatically in a correct way, but it's useful to check. So we're getting our column here as an attribute for the node, and we're gonna get it as numbers. So that's our column here, and then there's more columns that are also going to get in. In case the line is not, the rows are not split as we expect, we've seen it before with when importing the network, we can go to advanced options and check how the columns are delimited. In our case, we're delimiting on tab, and the file is using tabs, so it's split fine. If we split on space instead of tab, you see we're not getting things separated the way expected. So to get this problem fixed, we go back to tab, and we're once more fine. In most of the cases, Cytoscape is gonna figure it out on its own, but sometimes you've got to help it. The way to do it is to go to advanced options and see how columns should be split. Okay, so we've got the file ready to be imported. So now we click on OK, and as you see, our node table became now more interesting. We used to have just to share the name and name corresponding to the official names of the genes. Now we've got additional columns with numbers. Now those names are pretty meaningless. Those are formal ways of referring to a particular gene on a particular chromosome in Saccharomyces. They are very systematic, but they are pretty meaningless. To get something more reasonable, we can use another file that's in a directory. Let me find it. Let's go to my directory here, and I think I do have another file here. I can open it with Notepad, and here's another file which lists as before in one column those formal names of the genes, and the second column lists the name of the gene, the common name that sort of corresponds to whatever the gene. Those are more meaningful, more human readable versions of the gene names. In some cases, there's nothing meaningful available. This is probably because this was a gene discovered when the genome was sequenced and wasn't characterized at that time. So nothing is known. There was no, hasn't been discovered in any other way. So the meaningful name is not present. So then this formal name stays on. So here we've got another file to add to our Cytoscape network. So let's do it. Let's go to File, import once more table. Okay, now I did look, and this was the file, servici name. So let's select it here. Let's do open. Once more, we've got the import from table pop-up. We're getting those annotation to our network collection, so to all the network sitting that we're dealing with. Okay, it goes as a node table. Now splitting is fine. The first column is the key. Those are the formal ORF names. Second column are the human readable names. In this case, the file contained the header, contained the line with the titles for the columns. So Cytoscape figured it out and used those to name the columns, and you can see it in advanced options. It used the first line as column name, so that's correct. And then it's importing the actual values, actual annotation, starting from the first row after that. So we are fine here, and it figured out we are supposed to be splitting on top, so this looks fine. And now when you go on gene and click here, we're getting gene as an attribute, and it's a string. So that looks okay. So once we're ready to import, we just import, and now we've got additional column with the human readable name. The tables in Cytoscape work just like spreadsheets, so we can, for example, drag them around to move the column to wherever we prefer. So we can sort on a on a given column. We can, and it's either alphabetical in case of strings, or it's gonna be according to numerical value if the values in a column are numbers. So we can sort it this way. Now I've noticed that there's an extra node which is pretty uninformative. There's something unknown here. So let's check what this thing is. Okay, we can select a row, and to see where in a network it is, we can right click and then pick select nodes from selected rows. So now whatever is highlighted in a table is gonna be highlighted here in a network. And now to zoom on it, we can either painfully click on plus, or we can just fit selected and then zoom a little bit out. And it turns out that there's something strange in the file that doesn't look like it is. So we can get rid of it. As you noticed, if I hold control, I can drag the selected element. It can be either single node, or I can have more than, I can have, let's say, two. I can have two nodes selected, and then when I drag, then both are gonna be moving around. So this is the way to rearrange the networks by hand. Either we can drag a single node, we can select three nodes and move them around, or we can select an edge, and then if I'm careful enough, with edges it's tricky because it's very hard to click on them. So I can't do it, but you can drag the edges as well. Okay, and then going back to our strange things, I don't know what this thing is. It's probably glitch in a file. So to get rid of it, I can right click, and nothing happens, but I can I can select and I can go to edit, 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 and I can, where is it? And I can delete selected nodes and edges. Pressing delete also would work. I can select a node, press delete, and the node is gone, so we don't have the uninformative one here. Okay, so this is example how to manually edit edit the network, modify, make tiny corrections. Okay, so now we do have nodes with a bunch of information there. The What's useful about Cytoscape is that once we've got either nodes or edges annotated with the information, we can use it to actually decide how the network is shown. So let's do it first. Let's try to replace the formal name of the node with something which is human readable with the gene names here. To do this thing is we've got to go to style. There's a tab here called style, and this is the way, this is where the look of the network is determined. Initially, you've got some default settings according to one of the predefined types, predefined ways of network is shown. This is the default setup. There's more than one way to show things. This is the default selected. Now we can go to default on the background, and now we've got the same network just displayed in a different way. There's yet another one with another things shown. As you've noticed, the shape of the nodes is changing between the styles. The way the interactions are shown is changing. In this particular case, we do have a label shown for the edge. In this particular case, we don't. So all those elements of the graph displayed by the Cytoscape can be modified to your taste. The way it works is, the convenient way to do it is first to make yourself a new, to copy a style that we're starting with. Let's say, let's copy this default style and let's call it my style. Okay, so now I should have here the style I'm gonna play with without messing up the default. So let's select my style, and now I want to show, instead of the sticker here corresponding to the name of the node, I want to put the gene here. Okay, first I've got to find where it is specified that this is what's showing up as a label. Well, there's a label, okay, and it does have in a column called map, there's something here. When I click on it, it's gonna open a panel showing me that I'm mapping a column called name using a so-called pass-through mapping. So what it means is the name, the content of the name column here is taken and passed to be shown as a sticker here. If I want to use a different column to show as a label, I can click on the column in a column row and select the gene as a pass-through. And as you see, now we've got the values in a gene column shown as a label. We passed, this value is passing passing from the column directly to wherever it is displayed. This is so-called pass-through mapping. So this way I can pick any of the columns and show the content in a as a label. So here's one example of how we can modify the graph. So this starts to be more human readable. We can also use the values coming from experiment and show them on the network, for example, as the different shades, different colors of the nodes. Let's try to do this thing. Well, first we've got to find what is the color for the node. So this is fill color. At this moment, there's no mapping. There's only default blue color for the node. If we want to change all the default color to something else, we can just click here and make the nodes different. Let's make them green. Okay, and then if we do okay, then the nodes turned green. So this is the way to change default color for the node. Let's go back to blue of some sort. Okay. So now, what if we want to change the color depending on the value for a given node? Let's do it. Well, we can check the mapping here. Now let's say we want the gal 80, the second from the end column used. So let's select column. Let's get gal 80 RX here. So this is our column that we're gonna take the value from, and then mapping time it's gonna be continuous mapping. Okay, and here's a default setting with negative values getting progressively more blue, the positive values getting progressively more red. So this is something default. So now what we see is we see here three proteins that are overexpressed under given conditions compared to reference. We've got other proteins that with expression going down. So this starts to show us some experimental information overlaid on the network. If we don't like the colors, if we want to change the way the colors are assigned, we can click on the graph here on the gradient here, and now we can modify what's shown. Now how to do it? Okay, so we can double click on the triangle here, and we're gonna get the pop-up, and now it allows us to choose the color corresponding to the leftmost value, the lowest value, -3 or whatever. So let's make it let's change it for a moment. Let's make it slightly different. Okay, so this is the way to change the color here. Now here we can do the same thing and double click, and we can try to make it, let's say, purple. Okay, so this is the upper limit. We've got the center value corresponding to 0, and if we want to change it from white to, let's say, maybe a gray thing. We can make it gray, so then we can modify. We can also slide those things to decide where the gray color goes, so we can put it on 0 here. And we can also delete the handle, delete this value here, and then if we delete it, now we're gonna have a different gradient, now going from the green to purple, but not passing through a gray, but passing through some intermediate purple color. So so that's probably not good. So we can add another handle back, and then we can put it back to 0. Okay, and we can double click, make it gray of some sort. Okay, and here's our modified way the network is shown. So that's how to overlay numerical values on the top of a network using the background color for the or fill color for the label. For this particular experiment, we also do have the confidence or significance value here, so we can also show it in some way on the same graph. Let's say we take, let's say we'll use a border around the node to show how significant the color is. So let's try it. Okay, so we can go to border width. Well, first I'm gonna make the border black just in case. Okay, so it's gonna be black. And now for the so the color of the border is fixed, is always black, but now width by default is 0, but we're gonna try to map it. We're gonna go to column, and this is gonna be Gal80 significance. Okay, let's pick it here. Okay, so this is our column with the values that we want to use to draw the border. And now once more, we're gonna go to mapping type. We're gonna go to continuous. And now we've got some default guess, which may or may not be what we like. To my taste, this thing is too wide, so we don't see anything. So let's try to modify the mapping. We can once more click here. Now the mapping panel looks different because now we're mapping a numbers going between 0 and 1 to another set of numbers corresponding to the width of the outline of a label. 30 is apparently too much, so we can try to slide it a little bit down. Let's make it 15. I'm trying. Okay, and then it should go thinner here. So let's go this way. Okay, now this is actually not what we want. The significance is given as a p-values is given as the smaller the number, the more significant the changes. Okay, so we should actually flip it. So we should actually make 0 look like 15, and then we want to make numbers close to 1 going down. So let's make them down to about 3. If So that's our mapping here, and then we can do OK. And now we've got, oh, shared, oh, just a moment. I'm shared folder. Let's see, where is it? Where's my, okay, the shared folder is here. This is the one here. Copy. Okay, here it is. Hopefully it should work. Yeah. Okay, so that's the, so that's the network. That's the network that is now showing how much expression of a given gene changed in a given set of given condition here, and how significant this change is. And this is marked by the thickness of the border. So for this particular gene here, the change is presumably pretty high, pretty large, because the color is pretty intense, and significance is pretty high because the border is pretty wide. For this gene, the change is probably smaller in terms of expression level, but it is significant because the border is very wide. So here we've got two different, although related, pieces of information taken from experiment displayed in a overlaid on a network. So that's how it's done for let's say expression data. And now now we can go to the edge table, and now we've got here two different types of interactions. We've got either PPs, those are corresponding to protein interacting with another protein. We also have PDs, so this is protein interacting with DNA with the gene corresponding to another protein. So we've got two different types of interactions. We can show them in a different way in a graph. To do this thing, we've got to do we've got to set another mapping. To do it, we now go to edge. As you might have noticed, so far we are working with mappings for the node. So that's what we've got. What we've got so far. We've got border, the color of the border of a node. We've got the width. We've got the fill color that's mapped. We've got how high this thing is, and so forth. And this is all about the nodes. There's another set of parameters that we can set for the edges. Okay, and let's try to color code protein interactions and protein gene interactions in different colors. So we've got to find which of those parameters correspond to color of the line. Let's see where it is. There should be not label color. We want to have line, source, stroke, color unselected. This is gonna be the color of the node. I can convince myself that's the one by changing the default value to, let's say, to something horrible. Let's make it red. Okay. So yeah, that's our default color now for the edge. So let's go back and make it gray as a default. But now we want to set up a mapping, and we've got two values here, and I want to set a different color for PP then and different color for PD. Okay, so first I've got to decide which column I want to use. Okay, so I'm gonna use the column here called interaction, or shared interaction also would work. So let's pick up, this is the column. And now for the mapping, I'm gonna pick up now discrete mapping. Okay, this is another. So far we had a continuous mapping for colors, for where a number is converted into a range of colors. We did have a continuous mapping from number to number, where we're getting number corresponding to significance level, and this was converted, mapped into the number corresponding to the width of the border. So here we've got another case. We're taking discrete mapping, and now we've got two discrete values. This is where the name of the mapping coming from, that we're looking at discrete values coming from a given column. And then let's say for PP we can edit color, and we can decide that protein-protein interactions will be, let's say, green. Okay, so now you see we've got green lines corresponding to proteins interacting. So gal8 interacts with gal8, and gal8 interacts with gal1, and gal8 interacts with gal4. But now we can also say that for PDs, let's make it, I don't know, let's make it orange, for example. So now the orange lines correspond to protein interacting with DNA. So that's the interaction between presumably transcription factor and the gene that it's controlling. So those are those orange lines, and there's apparently quite a few of those. Now, with protein-protein interactions, the direction doesn't matter, because this is protein, they just stick to each other, and that's about it. With interaction between transcription factor and protein, and the gene it's controlling, it's useful to know who's who. It's not clear from the graph. So we can try to add additional mapping, which is gonna make this line here different at the end, so that there's gonna be direction. We're gonna be able to look at that. To set this thing, we can try to look for something which gives us the direction. So there's something conveniently named target arrow shape. So it's probably how the arrows at the ends of the at the target, so at the ending end of the arrow of the line are gonna be. So let's to go to target arrow shape, and so far there's nothing here. That's why there's nothing at the ends of the edges. So let's set up a mapping here, and let's go to a column. Once more, we're gonna use the same interaction as before, but now instead of using the value in the interaction column to control the color, we're gonna just put in, for one of those cases, we're gonna put an arrow there. So for protein we don't want anything, but from protein DNA we can decide that we want to put an arrow here. Okay, so now if we apply, now what we see is we do have arrows pointing from transcription factor to the gene it controls, and apparently that's this is apparently transcription factor controlling Gal2. This is transcription factor also controlling Gal2, and so forth. So now we can see different different transcription factors controlling different genes. So we can refine it. So far, we do have arrow. Why source arrow that we don't have control over color of the arrow, which is sort of ugly. But let's see, maybe we do. No, we don't have delta. We suddenly don't have. Suddenly we don't have control over the color of the arrow, so it's got to stay like that. I don't see any extra. Oh no, there should be target arrow. There should be paint source. There should be. We're missing target. Oh no, there is. Okay, so this is color actually. Okay, so let's select this one. Let's set up once more mapping. Once more, it's gonna be the same interaction column. Let's take mapping type. Let's make it discrete. For PP, we've got nothing. For PD, we can edit color and we can pick up the one we had before. So this is the orange thing. I think this guy, this was the recent one. So let's do OK. And now we've got arrows with the correct color. So here's not too aesthetic, but otherwise sort of informative way of coloring the network, so that we can see what are the changes in expression level in a given condition, and what the interactions are. Are those interactions between proteins? If yes, those are green, or if it's interaction between transcription factor and the gene it controls, then it's orange with an arrow pointing towards the controlled gene. Okay, so that's a recap on basic Cytoscape functionality. Most of the capabilities of Cytoscape that allow you to show networks are based on whatever you've seen so far. No matter how complicated the problem is, the general idea is to create annotation for either edges or interactions, and for nodes with whatever values we've got coming from experiment or from computations or wherever. And then those numbers can be used, or values that go in a column can be used to control different elements of the graph, so that you can visualize the values on the network. So that's how that's why Cytoscape is useful in general. Okay, so we'll make a break now for 10 minutes. If you've got any questions, ask. Just stop me, wave, do whatever, unmute yourself and complain, and I'll try to react. So anyway, for the moment, we've got a 5 minutes break to just stop for a moment. Okay, any questions? No questions. Dead silence.
Hi, Professor. I actually have a kind of a self-interested question.
Yeah.
So I was trying to import the data from a database called DrowID, which is for Drosophila, but I seem to be having trouble. It doesn't show up in the apps. Do you know anything about how to basically import interaction sets?
Yeah. Well, so far I didn't go into importing stuff here. Can you paste the link to the place you want to get interactions from in the chat so that I can?
Yes, I'll do that right now. Yeah. 1 second.
So far I went just through what you can do once you do have data. So this is the deal of a problem. That's right. It's the problem. The first part is how to get the droid. Oh, so this is another. Okay, import protein network Drosophila database not download. Okay, so then it means I've got to get this thing. Okay, so then I need app manager, and this is called droid.
And for some reason, it doesn't show up, at least for me, in the app manager.
Oh yeah, it doesn't show for me either. Okay. All apps. Okay, so let's see, what do they say? Well, they say no download available. Okay, so this is one hint. The problem is, let's see, what do they say here? Droid Sophia has its own.
So I just sent a second link from the DrowID website, and you can download a jar file.
Okay, so now
when I try to
yeah, so
I get an error when I try to use it.
Okay, can you get me link to the database also in a
yeah, I posted a second link.
Oh yeah, this is second one. Okay, Droid for Cytoscape version. Okay, so this is not gonna work, and this is typical problem with apps. So the way it works is Cytoscape is well maintained. Cytoscape has a large group of developers who actually modify, update this thing, and Cytoscape itself is pretty active in terms of development, adding new features. Now, apps are coming from third parties. Apps are not written by people who are maintaining Cytoscape. Those are separate programs written by different people according to whatever are requirements by Cytoscape, so that they can work inside Cytoscape. But those are things that are prepared by various people, and it's quite common that something that was written, as in your case, in 2012, is not
for 2.8.
Not maintained anymore.
I see.
So there was a major change in the internal design of Cytoscape between 2-point-something series and 3-point-something series, and apps that were written and provided as a jar for Cytoscape 2.8 will not work with Cytoscape 3. So that's why they don't show up. They don't show up there. The good part, though, is that most of the interactions are available in a species independent way. So it's in principle not necessary to use those species specific things to get your interactions in there. So I'm gonna go through this thing just after the break to show you how to get interactions in general.
Okay, great. Yeah, thank you. I appreciate it.
So, but it's good you mentioned this thing, because this is a very common problem that you've got about 200 250 or so apps. Okay, so it means there it's potentially 250 different research groups providing one app each, and the chance that some
Mm-hmm.
of them don't didn't update this thing or stop development, stop funding, stop lost interest is pretty high. So some of
Got
those
it.
will
Yeah.
not work. So it's sad situation. It happens. Not all of them. Not all of them work. I mean, I used to have something here that was for point two, 2 point something version of Cytoscape, which I didn't port to 3.0. So I'm also to be blamed. I had something here. I think. mm, I don't, and I think it disappeared because it wasn't ported. So that's common from the apps. Things to recognize it is that apps are third party things. So it also applies in terms of licenses. Cytoscape is free. Cytoscape is open source free program. You can get it, you can play with it, you can do whatever you want with it. Some of the apps do access data that's copyrighted or controlled by the providers of the app, and then sometimes you've got to sign a license or apply for a license to be able to use an app. So this is because each app is essentially a separate program written by a different entity to
Right.
group of people who are maintaining Cytoscape.
Yeah.
So that's something to be worth be aware of. So because of that, personally, I prefer to use as small number of apps as possible, because then I'm independent of those things going online, offline, and stopping to work when the Cytoscape is updated. So in some cases they are very useful, but in some cases they might leave you in a situation where you can't rerun your analysis because apps stop to work. So it's useful to be able to deal with the data in form of files and processing them with scripts, because then you're becoming less dependent on many third parties. So
Right.
that's how it goes. So any more questions? No questions. Okay, so let's continue. Let's pretend the break is over. So one thing to one useful custom to get yourself in is to save the state of the network, state of your work relatively often, because it's quite possible that something is gonna go wrong, or you modify things in a way that you can't revert back. So it's useful to have things saved every now and then to save the current state of Cytoscape, so-called session. You can always go to File and Save As, and let's call it my net1. Okay, and I'm saving it as a session here. And once it's saved, I can just let's quit it for a moment. Okay, so I'm just gonna shut down Cytoscape. Do you want to save? No, just quit. Okay, so now Cytoscape supposedly died. Okay, and now I can restart it. Okay, I can open it. It's gonna open in a moment. I mean, Cytoscape is big, so the startup takes time. Okay, and now I can go to File, Open Recent. This is what I used to. Oh yeah, this one is trying to get something. This is one of the plugins trying to talk to the server it's set up to talk to. Okay. Okay, so once more, File, Open Recent, and this is the recent one, or I can just go to Open, and then I've got mynet1.sys. This is a standard name, standard extension for the Cytoscape session files, and then I open, and I'm back to whatever stage I was at the time I saved it. So this is the way to save everything there, including layout and the way the networks are displayed and all the annotation in a table. So it's useful to save it relatively often, because then you're less prone to errors and things that you can't revert back because you removed something from the network, and then you can't reconstruct it in a painful way. So it's useful to save every now and then. So that's what we've got here. As an example of dealing with plugins, we can try the one that I asked you to install. This is the bingo guy here. Okay. This is a app or plugin that is trying to find out which gene annotation, gene ontology terms are overrepresented in a selected set of proteins, or selected set of genes, whatever we're dealing with. Okay, let's see how it works. So we've got to first give our bingo results a name. So let's call it my bingo 1. Okay, I'm gonna first, so now we've got to specify which proteins, which genes we're looking at. So let's start with all the proteins in the network. So I'm gonna select all. So to do this thing, I'm gonna go to Edit, to Select, and I'm gonna select all nodes and edges. Okay, so now everything turned yellow. So this is the set of all the proteins that are in the network that are going to be used. This is a subset of proteins in each. There's about, and we can check how many actually. There's about 330 proteins or genes listed in a network. He's got about 5,000-6,000 genes. So this is a subset of genes in the network here. So that's got selected. Okay, we're gonna look for overrepresentation of annotation of those genes relative to whole annotation. So relative to all the annotation available for all the proteins in yeast. So that's what we're looking for. Okay, we're gonna use standard settings. I'm just gonna make significance level higher. So I'm gonna go to 0.1, let's say. And the annotation that we're gonna look at is gene ontology, so-called biological process. Just as a side issue, to get you what those things are is let's go to Uniprot and see how what this gene ontology thing is, in case you don't know. So let's see, I'm gonna go to Uniprot. Uniprot is very, very useful. This is one-stop place where you can find a lot of information about proteins. If you don't know the site, I'm gonna paste you this thing in a chat and bookmark it. And this is one stop place to find information about any protein you're interested in. In most of the cases, there's gonna be something there, provided you can find a protein. So let's find a protein. Let's get, in our case, let's get gal AD, and let's make it gal AD from yeast. Okay, so there's whatever bunch of results, but I want gal ADs. This is the protein I'm going after. This is one of those things that were highlighted in purple here. So let's see what's there. So if I select the entry and then if I scroll down, we've got here annotation that information about the function of this protein. Okay, it comes in two or actually three flavors. Okay, one flavor, one part is molecular function. This is description what the protein does. Okay, in our case, this is a protein that binds to DNA. This is a protein that acts as a kinase inhibitor, and few other more or less cryptic terms that describe what a given protein does, in a sense of how does it interact with other molecules. Okay, the second part of this annotation is so-called biological process. This is where, in what network, in what context in a cell the protein works. So in our case, this is a protein that deals with galactose metabolism. Okay, this is a protein that modifies activity of transcription factors. Okay, this is a protein that is an inhibitor of kinase activity of some sort, and so forth. So those biological processes refer to function, in how the protein functions in a cell. The molecular function goes after the molecular details of how this function is performed. There's also a third part of gene ontology annotation, which tells you where in a cell a given protein is present. So those different terms. They are originally maintained by Gene Ontology Consortium, but Uniprot is getting those terms in. So this is one-stop place to find something about proteins. And apparently Bingo is also doing the same. Bingo is pulling those terms in for all the proteins in a network and compares how often they show up in a set of proteins selected for the analysis relative to all the annotations for all the proteins in the yeast. So that's what Bingo does. And that's where the terms are coming from. Going back to our window for the Bingo. So we've got the name the analysis is gonna be using. This is significance level. We're looking at gene ontology biological process. So those are terms related to where in what what process in the cell the proteins are taking part in. We're using as a reference all the proteins in the yeast. So let's see what happens. Okay, and we're dealing with Saccharomyces. So let's start bingo here. Select directory. Well, no, do I? No, I don't want to save it for a moment. Let's turn it off. Okay, so let's I uncheck this thing and let's start. Okay, now we've got suddenly a lot of windows popping up. Okay, so let's move this thing away for a moment. Okay, and now this is our initial network with all the proteins selected. Okay, now we've got another network, another or group of networks here, and here's the network. Now it looks different and it shows different things. In the case here, the network corresponds to individual genes or proteins and interactions between those. Okay, Bingo is using the network to show different gene ontology terms that were identified as overrepresented. So this is a network formed by the annotation terms. Okay, and they form a network because gene ontology is hierarchical. Let's try to find a piece that shows you this. Yeah, so let's pull something here. So here, 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 okay, and here, let's see, and yeah, so that's what I wanted to pull out. Here and here and here and here. So what we've got is we started from biological process. So this is what we're looking for. We're looking at gene ontology, biological process terms. Okay, and then among those there's one called response to stimulus. Okay, this is a very generic term that contains all the different stimuli that a cell can respond to. Now, among those, there's response to chemical stimulus. This is a narrower category, which is part of, or which is also response of stimulus, but more specific. And now there's even more specific term, which is response to inorganic substance. It's as opposed to organic compounds. And then even more specific is response to metal ion, and even more specific term is response to copper ion. So you see the gene ontology terms are arranged in a hierarchy from the very specific terms at the very bottom, and then when you go up, the terms are becoming more and more generic. And at the very top is one of those two or three categories: biological process or molecular function. That is the parent of all the terms. So that's why what you see in Bingo is essentially a graph showing the terms that are enriched and how they are related to each other. And the size of the dot corresponds to how strong the enrichment is. So so you can see what terms are popping up are overrepresented stronger than others. So here's the network that Bingo returns. It also returned a table with the same terms. Let's look at it. So here's a table. we do have the terms. Okay, they are now ordered by p-value. They are ordered by how strong enrichment is. The p-value technically tells you how likely it is to get as many proteins with a given term in our set by drawing them by random chance from all the genes in yeast. So if, let's say, there's a term that is used to annotate 10 proteins in yeast, and if in our subset 9 out of those 10 proteins are present, then drawing 9 out of 10 proteins when we've got 5,000 other proteins is in a genome is pretty unlikely. So if we've got 9 of those, probability is very low. So then this p-value is very low. It means that enrichment is very strong. So let's say it this way in semi technical terms. So we've got a bunch of terms sorted by p-value, and we've got also genes listed here corresponding to individual that are annotated with the given term. So if we pick up something, let's say one of them, whatever it is here, if we pick up one or more, if we set the checkboxes here, and then we can select nodes. And now if we go back to the network here, I'm gonna unmask this thing, and now let's see if I select nodes. Now I've got a subset of nodes selected that are annotated with a given term. As you can see, this doesn't look too informative. The reason is we picked up all the proteins that are in a network, okay? And this is not too specific. Network is relatively large. It's composed of quite many proteins that might not be related to each other by function. They will have different functions. So there's not going to be any particularly strong enrichment with a given term. But what we can do is we can rerun the analysis, but we can limit the set of the proteins to proteins that are highly enriched in our conditions that we marked here. So let's try to do it. So first, let's select the nodes that are overexpressed in our conditions. Let's say this Gal80. To do these things, I'm going to node table. I'm gonna sort it by the column with the expression. Okay, so this is under expressed. I'm gonna go to overexpressed by clicking once more at the top. So now let's pick up, I don't know, let's pick up maybe let's pick up, let's say how many, let's select down to, okay, let's go down to, let's start from 3 all the way down to 0.5. Let's go here. So this is gonna be our last one. So with control and left click, not control, with shift and left click, I can select the range in a table. So now I've selected all the nodes with expression log ratio higher than 0.5. So here's 0.5. And now if I want to change the selection in a table into selection in a network, I can right click and then I can select the nodes from the selected rows. Okay, so now I've selected 47 apparently nodes that now are scattered somewhere throughout the network. Okay, so let's use this set of proteins as our input to bingo. Okay, so I'm gonna kill this thing. I'm gonna take once more and go to bingo here. Now I'm gonna call it cluster 2, my bingo 2. Now once more, I'm taking nodes selected in the network. Okay, so now there are only 47 instead of 300 something. Okay, and those are 47 that are overexpressed to largest extent in our data set. Okay, once more, it's gonna be 0.1, 0.01 significance. I'm looking at biological process Saccharomyces. I'm not gonna save anything. Okay, and let's start bingo once more. Okay, so now we've got another set of results from bingo. Now there's less nodes, less terms got overexpressed. But if we look at what they are, now the first two are corresponding to galactose catabolic process, galactose metabolic. There's another one related to galactose. So apparently our network is somehow related. Our condition, our expression data somehow identified proteins that are related to galactose metabolism. This is actually true. The set corresponds to an experiment where the GAL80 protein was removed from the cells. The gene was knocked out, and then the expression of genes were compared with the galactose present. So this is a ratio of gene expression in the presence of galactose with the GAL80 present or GAL80 absent. So that's what the data set is. GAL80 is a gene that controls expression of genes related to galactose metabolism, and then when it's removed, then the cells will react to galactose in a different way than when the gene is present. So that's why genes related to galactose metabolism are perturbed most. And we've identified that those are actually that's what's going on, because the genes that were identified as overexpressed are annotated as related to galactose. So that's what bingo tells us. And here's here's the set of terms, and now we can pick up let's say now all those galactose related terms. Let's pick up those three guys here, and then let's go back to the initial network. Let's unselect things. Okay, and now let's select nodes once more that are corresponding to those galactose metabolism, whatever they are. And now if we select those and look what's going on here, now if we zoom, now as you can see, we picked up quite a few of the genes that are known to be related to galactose metabolism. So In this particular case, it's just demonstration how the analysis works with things that we know the function of. But you can imagine that that similar approach is used for a setup where you've got a protein of unknown function that you want to identify what it does. So you remove it, you look at how expression changed in the presence and absence of the protein in a cells, and then you identify a group of proteins that are overexpressed, and then you take those and run bingo analysis. You run the enrichment analysis on those, and you pull out terms that direct you towards certain biological process. You might also, as here, map those genes on an interaction network, and you might observe that they show up within the same region of a network. So this is another hint that this is a group of related genes that are doing some particular function working within one particular network. So that's one of the ways of trying to analyze, look at protein function, and trying to identify what different proteins do and how they work in a cell. So as you can see, we picked up just a few proteins that are localized to one tiny region of a bigger network. So this is often the case, that proteins that are work together in the same pathway, they often interact with each other. So when they are identified and mapped on interaction network, they show up in one small region as a cluster within interaction networks. And that's why looking at those things is, yeah, as graphs is useful, because it's easier to find those things by just looking at the networks rather than by looking at the list of things. This is less informative than graphical display. Okay, so far we were working with a God-given 2 or 3 files that came out of nowhere. Essentially, it's the file where the expression data came from is most likely coming from one particular experiment. This is something which is generated during your research. This is what's coming out of microarray data or mass spec data, or whatever experiment happened to be. And this is generally prepared in-house. The problem is with the file that generated the interactions. This is more tricky. Because you've got to get it from somewhere. Typically, you're not going to be maintaining a list of all the interactions in between all the proteins in the organism you're interested in. You might know something about the pathway you're working on, but you're not going to know anything else. So the questions where the interactions are coming from. So let's make a 5-minute break, and we'll finish up by trying to pull in interactions into Cytoscape from outside, not as a file. So let's make another 5-minute worth of break. So, and are there any questions? Once more, if there are any questions, ask. Okay. Horribly quiet. So okay, so I'll become quiet and we make a break till, let's say, 11:05. sorry. Let's
Hi, Professor. Sorry for interrupting, but did you mean to mute yourself? I think that you're muted.
Yes, I'm muted, but I'm not talking. So
Oh, gotcha.
I'm just playing with whatever's on the screen.
Okay.
Okay, so now. Okay. Okay. Okay, so let's go on. Okay, so out of curiosity, I just just going for a moment back to bingo. Let's go to the table here. So those are those three terms: galactose, galactose, galactose. So I just selected those. Now I click on select nodes to select those. And as you can see, we picked up 1, 2, 3, 4, 5, 6, 4, 7 genes annotated with those three terms I've picked up. Okay, those are all the guys here. I just zoomed on selected nodes, and that's all I can see. So it means outside of the window here, there's nothing else. So there's no other genes identified as annotated with galactose related terms. Okay, how many there is total? Okay, we can check it by just clicking outside so that nothing is selected. Now I've got the node table showing all the proteins there. I can now sort it by gene name, and I can scroll down and find all the GAL genes. Okay, so there's GAL1 is the first one, last one is GAL80. So once more shift and GAL80 selects me its entire block, and now I can right click and select nodes from selected rows, and I've picked up the GAL genes here. Is there anything else where I can click on fit selected to see if there's something outside that I'm missing now? No. So apparently those are the genes in the network that are known to be related to galactose metabolism. That's where the names are coming from. Out of those, everyone but this GAL11 were annotated with galactose with those three terms. It could be because the annotation for GAL11 is obsolete. It could be there's another galactose term that Bingo didn't identify as enriched. This will require more digging into annotations. But the point is that not all the genes might be picked up, but they might be still related because they are sitting and interacting with the group of genes that was identified as working in certain pathway. So this GAL11 working in a galactose pathway is identified by so-called guild by association. This GAL11 genes binds to GAL4 protein and its transcription factor controlling other galactose genes. So it's probably also related in function to to galactose metabolism. So this is another way why looking at the networks is useful. If we find a protein that we don't know what it does, but it happens to be controlling, or it happens to be controlled by genes that we know what they do, or it happens to interact with a gene that we know what it does, it means that it's likely candidate for functioning in the same pathway. So this is another way of utilizing the graphs like this. Okay, where the graphs are coming from? So far, this one came from a mysterious file that I pulled out of the ceiling. How to get to data, the interaction data. Okay, so let's wipe wipe out everything, but before that I'm gonna save it. So I'm gonna save it as, let's call it mynet2, and let's save it. This way I'd be able to retrieve whatever we've got here. Okay, so that's saved. So now let's do file, and or we can actually well, we can do file, and let's let's do it this way. Destroy. Okay, destroy. To remove a group of networks, I can do destroy. Okay, and I'm gonna do here once more destroy. Okay, so now we've got the blank cytoscape. Okay, where to get interactions from? The standard, the built-in way of getting interactions is using so-called psychic. This icon is gonna be present all the time, even without installing any apps. I do have installed string, and I do have installed, yeah, I do have installed string and GenMania, so there are extra options showing up here, but by default, this psychic icon is gonna be present, and this provides us with the field to enter search terms that that we will use to find interactions. So let's try with the genes we had before. We had, let's say we had gal AD, we had gal4, let's say, and let's see what happens. So I just gave it two gene names, and let's search. Okay, the Cytoscape talked to servers, or to a server that provide to servers that provide access to interactions from different databases, and initially it returns a list of databases and what was found there. Okay, let's see what's here. Well, we've got a lot of things which I don't like, because this is gonna be a lot of junk. But let's, well, so let's modify it. Let's try gal AD only and see what happens. Okay, that's better. Okay, so let's look for interactions of a gal AD protein or gene. Okay, and I'm gonna pick up the IMAX. This is a group of consortium of databases that curate interaction data in a uniform way and provide access to those through one of the psychic servers. I'll go with IMAX. Okay, to start with, I select that I want to import interactions from there, and then I'm gonna import. Okay, and here I want to close. Okay, and here what we're getting. Okay, now we're getting a network that looks funny, but does have some proteins there. Now, as identifier of the node, we're getting identifier of the protein in a Uniprot. If you look at what this P043 something is, this happens to be Gal80. This is the human readable version, okay? But the identifier that's used, if I take it, I happen to know that this is identifier that used by wrong field. This is identifier used by Uniprot. See, this is exactly the same identifier here. In general, IMAX servers, in general, psychic servers will get you identifiers of the. This is not. Will get you as a identifier of a protein the entry in a the record identifier in Uniprot. So this is different from whatever you've seen before. What you've seen before, there was a systematic ORF name in yeast used as a protein identifier. IMAX is going to return Uniprots as node identifiers plus extra annotation which might contain those standard ORF names. It returns more things. Okay, it returns the taxon ID, the formal identifier of the species the protein is coming from. It also gives you a name of the species. Okay, so this way we know that those are mostly yeast proteins, but not all the time. So there are other things present, either not from yeast or not even proteins. Okay, this is not a protein. This is a small molecule that happens to be participating in one of the interactions. Some of the IMAX servers, some of the IMAX databases do curate to provide information about interactions between protein and a cofactor protein and a molecule that the protein recognizes. Okay, this happens to be galactose. As I've noticed here, there's a name which suggests it's a galactose. Well, it's quite appropriate for a network of proteins dealing with galactose metabolism. Okay, what else do we have? We do have many other alternative names. Okay, in various formats. We're getting various cross references in the form of a string. So those are cross references to things like gene ontology terms. Okay, apart from text representation here, there's also identifier. So there's RNA polymerase II activating blah blah blah. This is a gene ontology term with, this is the human readable description, and this is the identifier of this term. So there are lists of those, a lot of stuff coming in here. There's more annotations. There's a lot of stuff that it's pretty cryptic. I don't know all those things. It's a lot of information. Is pretty disorganized, but it is there, and that's what you're getting for individual proteins or small molecules. So this happens to be no, which one is it? Oh, I don't see it. Okay, let's see what is this one. Let's select no. Yeah, so here, so this is apparently galactose. Okay, so it once more, it's pretty pretty complicated in a sense. There's a lot of information there that's pulled in, and takes patience and a lot of looking around to figure out what's there. But information is there, and there's a lot of it. Same thing goes for edges. Same thing goes for individual interactions. One thing to notice is that, let's look at these two proteins here. Okay, so we've got three edges connecting two proteins. Okay, if we select those, okay, let's try to select those three. What we're gonna see. Okay, let's see what's the annotation for those three. Okay, I selected three nodes. I'm looking at the edge table. So now I do have information that came from Psychic, from IMEX consortium about interactions between those two proteins. Okay, so the name is gonna look different. Now, instead of interaction type here, there's apparently identifier of the edge that came from the database that curated the data. So this is different than whatever you've seen before. But at the beginning, at the end, there's a protein identifier at one end of the interaction and the protein identifier at the other end. So those are two identifiers at the ends. We do have a reference to the paper that the given interaction was reported. Okay, somewhere down there's a PubMed ID. So this is identifier of this paper. See, once more, it's not organized in a perfect way. You've got the author showing up here, but then pub ID is sitting somewhere here. So it's sort of complicated to put those things together. So it's not clean, let's say it this way. What else do we get? We're getting so-called primary interaction type. This is information. This is information of what was detected in a given experiment. As you see, we've got three edges here. Each one is corresponding to separate experiment. This is because each IMEX, each Psychic record that's returned by querying for GAL-AD, each record corresponds, each interaction corresponds to information about one particular experiment that identified that two proteins or two molecules interact. There were apparently three different experiments, each demonstrating that those two proteins interact, and each one generated separate edge and separate interaction. And each of those is determined, presumably, possibly by a different method, and the experiment might have provided different type of interaction. Two of those apparently provided information that those two proteins are in direct contact, that two molecules touch each other, and experiment was able to determine that. The third one, in third experiment, it was only identified that those two proteins are in the same physical complex, but they might be bridged by some intervening molecule out there. So this is a case where protein A binds to protein B binds to protein C. So then A and C are in the same physical complex, but they don't directly interact. So in those cases, interactions will be annotated as physical association. So that's why we've got the different types for the same pair of proteins. It's a reflection of how accurate the experiment was, how informative the experiment was. So apparently in 2 cases it was possible to identify proteins as interacting directly. In the third one, it was possible that there was something else bridging. So that's how it was reported in the record. So here we've got the interaction type. This is different than interaction type that we've seen here. This is not the convention is not doesn't apply for IMAX records. Okay, so next what we've got is, you might wonder what are the different experiments that were used to identify that those two proteins interact, and we've got 3 entries. One was protein complementation assay, which my guess is was most likely is to hybrid, one of the version of protein complementation assay. So that's one experiment. There was a pull down performed, and this is second experiment. And then there was a cross-linking experiment done, and all those 3 different methods identified as those 2 proteins interacting with each other. So this is once more to notice that those multiple edges connecting proteins, when the interactions are coming from the IMAX, from Psychic, correspond to separate experiments. This is essentially the same interaction, but identified in three different independent ways. What else do we have? We've got confidence score, which tells something about how reliable the data is. The confidence score goes between 0 and 1. The higher the number, the better. It's generated in a pretty arbitrary way, so it's useful, but it's very hard to set actual threshold, and to assign a meaning to particular value. The thing to know is that the closer to 1 it is, the more reliable the experimental data is. And then there is a bunch of other fields related to individual experiments that were performed, and what was the role of the protein in given experiment. Was it modified? There's entire slew of those. We'll go over those tomorrow, and probably it would be good if you read through this PowerPoint, click through the PowerPoint that I dropped in. I dropped here. If you click through this thing, there's a brief outline and few examples of different methods that are identifying that can be used to identify protein-protein interactions. There's some information about interaction type and distinguishing between direct and the physical interactions, and it will pop up to go tomorrow. So it might be useful for you to click through those things. If you did take the intro to Cytoscape, I spent I think more than a day going over details like that last quarter. So if you didn't participate, just try to click through the file and see what's there. It will help tomorrow. Okay, so those are all the extra information, extra data coming from the Psychic. So here we've got the opposite situation. With the initial file, we had only protein interacts with protein interacts with DNA, and nothing else. Here we've got diarrhea. Here we've got a lot of stuff that is coming in, and the problem is deciding on what to keep, what to remove, what how to deal with that. So this is the issue with when importing things from Psychic. You've seen the case of overabundance of stuff by adding um, when I added just uh, gal gal 4 when I added, and when I'm looking for two proteins or two genes instead of one, when I try to search for that, now instead of few things, IMAX is giving me half a million of entries. Okay, this is presumably because gal4 might exist in different species. So what if I do east and see what happens? Okay, that's better. No, not better. We're getting even more. So let's try to do like this. Let's try it this way. Let's try gal AD or and let's see if this thing works. Ah, now we're better. Okay, so the lesson learned is that whatever we put here, whatever collection of terms we put here, initially comes as gal 80 or gal 4 or gal 2 and so forth. So the format of the query to the Psychic is essentially a list of keywords. So this is like in Google, that you're providing a list of keywords to look for, except that those terms are connected by or. So a record is gonna match if it mentions gal 80 or mentions gal 4 or mentions gal 2. It happens that gal 4 is a very popular name not only in the yeast but in many other organisms. So by putting gal 4, I'm pulling half a million of entries from who knows where. So to limit it, I'm gonna actually ask for uh, gonna explicitly ask for gal 84 or gal 2, but I'm gonna put it in brackets, and I'm gonna require that this is also annotated and it's coming from yeast, or the interaction record mentions yeast. So this way I can now search. Okay, and now IMAX is giving me 126 records that match yeast and one of those three gene names. So now I can pick just what I want to get here, and then I'm gonna import, and here's what I've got now. This part is similar to we had before. That's right. This is what we had before when looking only for gal 80. We got less things because there's less records corresponding to gal 80 interactions, but some of them, as you've seen before, involve non-yeast proteins. So here's some strange viral protein, I think, of some sort. Okay, here's galactose. Okay, in the case here, we looked for gal 80 or gal 4 or gal 2, but we required that the record mentions yeast. So now we've got this extra stuff here plus many other proteins. So this way we can pull in interactions from a given species for one or more interactions. So this addresses the question about the C. elegans or Drosophila or whatever other organism is. one can look for interactions from a given species for a given gene or a group of gene by specifying a query like this in a psychic search field, and then you're gonna get interactions returned from the databases that deal with interactions that make the records public and visible through the psychic interface. Let's rerun this once more. Okay, just to show you what's showing up here. So this window is gonna show you a list of different servers, that different databases that provide information about interactions. In a PowerPoint file I left, I dropped on a Google Drive. There's a short overview of different resources that deal with interactions. Some of them do show up here. Personally, I do prefer to work with IMAX. One of the reasons is that the database we're running at UCLA is also contributing to IMAX, so it's self advertisement. But besides that, I know details how the interactions are processed, how interactions are curated, how the records are curated, and it's a reasonable balance between providing enough information that's detailed enough to be able to interpret those in a reasonable way. It's not too superficial, and from past experience, I think that the quality of data is pretty reasonable. Intact is one of the databases within the IMAX. They curate things outside of whatever IMAX covers, so that's why Intact gives you a little bit more interactions there. Mint is another IMAX database, so those 51s are probably contained within whatever IMAX search returns. And now there's many other things. One of the large resources is Biogrid. It's mentioned in a PowerPoint. They've got a lot of information, a lot of interactions listed. They are much bigger than IMAX in terms of number of interactions reported, but annotation is pretty superficial, and sometimes it's not detailed enough to get the information that you're interested in. Okay, and then there's more things depending on what's the query, depending on what you're asking for. One, two, or more of those resources will have records to be reported. Some of them are computational, so Intact, IMAX, Mint, Biogrid, InnateDB, and few others are resources that return information about experiments, about interactions determined in experiments. Some of those resources might return predictions. This Gene Mania is gonna return mostly predictions. Another source that there's no string here. String used to be here. So out of those things, Gene Mania is returning predictions. So this is something to be careful, because some of those resources might not return what you're interested in. But anyway, this is the place to. Those are the different resources that provide information about interactions that you can get through the psychic search. So let's cancel this thing, and then let's, in the last 5 minutes, let's try to clean this interaction network a little bit. So let's first, I'm gonna rotate it a little bit. Let's say this way. Okay, and let's select Okay, so one thing which is or is not useful is those multiple experiments identifying the same interaction. By the way, the interaction that starts on a given protein and goes back into it means that this protein forms oligomers. There's quite a few of cases like that. So this is protein that forms oligomers. This is protein that forms oligomers. This one is not, but this one does. So some of those things do have loops corresponding to oligomers, but otherwise there are edges that are multiple edges connecting the same protein. So to get rid of those, we can try to go and there's gonna be, well, so one version is to go to layout and we can bundle edges. Okay, and no, we're not gonna bundle edges. No, we're gonna just do the simplest thing. We can go to edit and we can go and remove duplicate edges. Okay, so now we've got those two networks, so let's use both. And now we're gonna ignore edge direction. In some of the cases, interaction might be reporting as starting here and ending here, and then another experiment might be annotated as starting here and ending here. So if we want to collapse to treat those edges in the same way, then we've got to ignore the edge direction, okay? But we're gonna create an edge table column with number of duplicated edges. What it means is for the edge connect, for this homooligomer, we're gonna have a column with a value 1, 2, 3, 4, 5, 6, 7, which is going to tell us in how many experiments a given interaction was identified. So interaction between the node here and the node here will have a value 3 because there are three edges there. So let's create this edge table column with the number. Okay, so let's check both things here. Let's decide we want the second guy. So this is 4405. So this is the one that I want to collapse. And now let's go here and do okay. Okay, and now we lost 44 edges. And we still didn't collapse this thing because we only applied it to the edges that connect to different proteins. So we've got to go back to edit. We've got to repeat the process for self loops. Okay. And we're, no, why it didn't? It should collapse. It should collapse the, that's mysterious. I don't know why. This might be a bug. This should have collapsed. So let's get rid of self edges anyway. Let's destroy, just remove self loops. Okay, and I'm gonna just remove it. Okay, and now the loops are gone. Okay, so now we cleaned up this thing a little bit. We can now fix the layout. We can go to layout and try organic, let's say this way. Okay, and here's the network that got pulled in and got cleaned up a little bit. One thing to notice is that now when you look at the edge table, Let's see. We've lost some of the information about the interaction type. As you remember, for the interaction here, we had three edges. Okay, we've got three edges, and the, where is it? And the interaction type is different between those three experiments. When those three edges are collapsed into one, only one type is gonna be left and the other two will disappear. So the operation which made the network cleaner removed some of the interactions here. If we look at the edge here, see now there's only physical association, even though the number of, even though let's see, even though there were 9 experiments demonstrating this interaction, and it's most likely some of them had different interaction types than the others. So this operation where we edit and remove duplicated edges is lossy. Some of the information is lost, and there's no way to control it directly from the cytoscape from clicking. Still, as long as we don't care about interaction types, or if we do filtering somehow beforehand, before then the removal of duplicated edges might be okay. It depends on application, and it requires little bit of thinking, in a sense that we've got to know what type of interactions we're trying to look at, and how to handle those things. So in simple cases, clicking in Cytoscape will work. In some other cases, it goes down to writing scripts that modify things. But anyway, that's the simplest way of simplifying the output coming from Scikit. So I'm gonna stop now. Okay. If you want to practice, I would try, for example, to, let's open the previous thing. Let's try, let's open current session. Okay, I'm gonna get this guy. Okay, so if you want to practice, just try to, instead of using gal 80 to color the network, and to color the nodes, and to set the borders, try to do the same thing, but for either gal 4 or gal 1. And once you've got it, you can also try to run bingo and see how the enrichment goes, and does it look the same or does it look different than for gal 80. So my guess is that in one of those cases, you're gonna see a similar pattern of enrichment in the terms as for gal 80. In the other case, it's not gonna look different, and there's actually some sense to it. So if you play with it, try to ponder why there are differences there. Okay, I'll drop this thing. I'll drop this file this network, the mainnet tool on the Google Drive. So you'd be able to start with this network in case you didn't click through and didn't have a chance to get that far with the playing with network. So that's it for today. Tomorrow we'll try to try alternative way of getting data into Cytoscape by writing scripts in Python. I mean, initially it's gonna be something very simple, just to get you going with getting some information in from just Uniprot, and then we'll go after interactions. It's gonna be mostly interactive, going with through Spider, and I'll have a file in Spider for you to follow whatever I'm doing. So it would be good for you to install Anaconda. As I mentioned before, Anaconda is sitting here. So if you go to anaconda.com, then you go to products, individual, and then down here there's a download, and just get one of those three things and download it. The files are sort of long, so depending on your network, the large also depend on your network connection. It might take a while, so it would make sense if you do it before showing up tomorrow. Okay, any questions? That silence, that's scary. Okay, so if there's no question, we're done for today. I'm dropping the file with the Galactose network on the Google Drive, and I'm stopping the recording, which means Eloy should be able to drop the recording on the Google Box. Okay, thank you for participation today. Stop recording. Do you want